Every spring, under a full moon, the beaches of the American Atlantic coast come alive. Creatures that predate dinosaurs and even trees haul themselves out of the surf. They have been making this exact journey for 450 million years. Today, human survival relies on them. Their unique biology serves as an invisible, mandatory quality control test for the modern medical system. If you have ever received a medical implant, an IV drip, or any kind of injectable medication, it was tested using a compound found inside this animal. Without their biological contribution to screen out dangerous bacterial toxins, the safety infrastructure of global healthcare would fail overnight. Despite the name, the horseshoe crab is not actually a crab. It belongs to an ancient lineage closely related to spiders and scorpions, protected by a smooth, armored dome and a long, rigid tail. Beneath that shell runs an unusual defense mechanism. Its blood is brilliant blue, a result of using copper instead of iron to carry oxygen through its body. That blue blood contains a specialized substance called limulus amoebocyte lysate, or LAL. When LAL encounters bacterial contamination, it reacts instantly by physically clotting around the foreign matter, trapping it completely. In the 1970s, the pharmaceutical industry adopted this mechanism to test all injectables. It proved so reliable that it became the global standard for detecting microscopic, dangerous bacteria in sterilized equipment and medicines. As a result, our most advanced medical technologies remain tethered to the physical extraction of fluids from a wild, prehistoric sea creature. To meet global medical demand, the pharmaceutical industry captures approximately 500,000 horseshoe crabs from the ocean every year. These live animals are transported to industrial facilities where technicians puncture their hearts, draining up to 30% of their total blood volume before shipping them back to the water. The process is intensely stressful. Research shows that up to 30% of the blood crabs do not survive the procedure or die shortly after release. Treating a finite wild population as an inexhaustible manufacturing component builds a severe vulnerability straight into our medical supply chain. Most of this harvesting happens along the eastern seaboard of North America, stretching from Maine down to the Gulf of Mexico. The epicenter is Delaware Bay. Every May and June, over 1 million individuals crowd these beaches to spawn, with large females dragging males out of the surf to lay up to 80,000 eggs each. Right as this happens, a small migratory bird called the red knot arrives. These birds are in the middle of a brutal flight from Argentina all the way to the Arctic. Delaware Bay is their only critical refueling stop. To survive the remainder of the journey, the red knot has just 2 weeks to double its body weight, eating almost entirely horseshoe crab eggs. Because the medical harvest reduces the number of spawning crabs, there are fewer eggs in the sand. Without enough food, the red knot populations collapse within a single breeding season. Our requirement for sterile medical equipment drives a ripple effect that spans continents, linking routine human healthcare directly to the survival of migratory birds. This chart shows the historical population baseline of horseshoe crabs before the 1970s. Combined with decades of harvesting for bait, intense biomedical demand led the IUCN to list the species as vulnerable. In specific regions, localized populations are depleted by over 90%. A viable solution exists. Since the early 2000s, scientists have produced recombinant factor C, or RFC, a fully synthetic laboratory alternative that perfectly replicates the crab's natural clotting compound. Several European pharmaceutical companies have already completely transitioned to this lab-grown alternative. In the United States, however, full adoption remains tangled in regulatory reviews and delayed approvals. We have the scientific capability to end the harvest tomorrow. The primary barrier keeping the horseshoe crab in danger is human bureaucratic inertia. To monitor these fragile populations without causing further stress, marine researchers are now testing environmental DNA. By sampling coastal water, they can measure the genetic material shed naturally by the crabs and track their numbers completely hands-off. This is an animal that survived the asteroid that killed the dinosaurs, enduring every global mass extinction event in Earth's history. Yet, it now faces its greatest threat from human industry. Securing the medical supply chain and ecosystems that rely on this creature requires adopting synthetic alternatives, leaving them in the ocean where they belong.